connection to the other side, wherever this is feeding out to. So today we have 3 master's prospectus talks, or prospectus presentations. All 3 students are in my physical oceanography class, so I'm happy to introduce them. We have Kim leading off, then we're going to have Ed and Merritt. I don't know if you guys...
We haven't.
Yeah, so we'll figure it out as we go. We have these 3 students, and we have a lot of oysters and a lot of fish today, so Kim is leading off with an oyster talk, or oyster perspectives.
All right, good afternoon. I'm Kimberly Peter, and I will be presenting my master's prospectus on eastern oysters as retrospective bioindicators of oil contamination. We all know the Deepwater Horizon oil spill, which occurred in 2010, had huge impacts on the marine environment. The spill released about 4.9 million barrels of crude oil over an 86-day period. The oil entered the marine environment and immediately began to weather through a variety of processes, including evaporation, dissolution, sedimentation, photo, and microbial degradation. The weathering process removes the low molecular weight hydrocarbons, leaving behind the heavier, more toxic components. This turns oil from the consistency you probably think of as oil to a creamy caramel, and finally to tar balls like you might find washed up on the local beaches. Trace metals are bound to the hydrocarbon structure of oil, and oysters can accumulate these trace metals into their shells and soft tissues. Shells provide a historical record because it is laid down in layers as the oyster grows. Tissues are quickly metabolized and provide a record of only the most recently assimilated elements. As oysters feed, they can select particles they don't want to consume and remove these as pseudofeces. Particles that are consumed pass through the gut and are assimilated into the tissue and shells, as previously described, or they can be combined with catabolic products and removed as feces. The assimilation efficiency of trace metals, such as those bound to oil, are both elemental and species specific. These graphs show time on the x-axis and the percent of 6 different trace metals retained in the soft tissue of 4 different species of bivalves on the y-axis. So each of the graphs show 4 species of bivalves, and you can see that the assimilation efficiency is species specific. But what I want to specifically draw your attention to is the elemental specificity between individual oysters. My study species is the eastern oyster, and I have highlighted this with red lines. And you can see variations among the different elements. For example, cobalt here has low assimilation efficiency with, after about 16 days, only about 10% is retained within the soft tissues. Conversely, zinc in this bottom graph has high retention with, after about 14 days, roughly 80% remains in the soft tissues. The bioaccumulation of elements can be affected by environmental factors. For example, salinity is a major factor that affects the northern Gulf of Mexico because of a large influx of fresh water from the Mississippi, Atchafalaya, and Mobile River systems, which creates a dynamic environment that can affect the assimilation of trace metals. These graphs show salinity on the x-axis and the concentration of 3 different trace metals on the y-axis. Again, you can see species variation among these 2 species of saltwater clams, and you can see variation among the trace metals. But you can also see variation within a single species and a single trace metal with salinity. For example, this saltwater clam with the open circle here has a cadmium concentration of roughly 0.15 when the salinity is 5, but that drops to about 0.05 when the salinity raises to 30. Another factor that can affect trace metal assimilation in oysters is high background levels. Haley created this map of the water treatment plants in the Mobile Bay watershed. Effluent from these treatment plants drains into the Mobile Bay and likely causes high background levels of trace metals in our local estuary. Copper and lead are elements typically associated with wastewater. Stable isotope analysis can be used for dietary reconstruction. Here you can see that the carbon-13 signature of marine algae is roughly negative 21, whereas the oil signature is roughly negative 27. But freshwater can make it difficult to distinguish the oil signature using bulk stable isotope analysis because the carbon freshwater signature is similar to the oil signature, both reflecting deltaic sources, which is carbon that originated from terrestrial sources. Compound-specific stable isotope analysis might help to resolve this confounding factor. Compound-specific stable isotope analysis is an emerging field that can be used to further identify sources by analyzing specific analytes. The Mussel Watch program was established in 1986 and monitors 300 sites across the United States. They use mussels and oysters as bioindicators for various pollutants, including trace metals. The Mussel Watch program analyzes soft tissues, which requires frequent sampling because of high metabolic turnover. Shells may provide a parallel analytical method to complement the soft tissue monitoring projects and allow for long-term retrospective assessment of contaminant exposure not possible with soft tissues. The purpose of my research is to complement the Mussel Watch program by determining if eastern oyster shells provide a record of oil exposure through time and can be used to determine both historical and current exposure of oil not possible with soft tissues. The first chapter of my project is to use trace metals in oyster shells and soft tissues to detect oil contamination in the marine environment. I completed this with a controlled laboratory study where I had 20 tanks and 6 different treatment types. The first 3 are the Macondo crude oil, or the Macondo oil, which is the oil from the Deepwater Horizon oil spill. I have crude oil, weathered oil from the surface, and highly weathered tar from the local beaches. The Macondo oil is known to have less of a trace metal signature than most other oils, and for this reason, I have included an oil standard that was spiked with vanadium, copper, and zinc. My 5th treatment was to expose oysters to water from the Mobile River, which is a highly industrialized region that likely has high background levels and potentially oil and trace metals. Finally, my control was filtered seawater. The second objective is to determine if the concentration of oil affects the chemical signature. I created a saturated, high-energy, water-accommodated fraction and exposed oysters to either 0.3% or 3%. I have shaded my tanks here to indicate this, with the lower concentration of 0.3% shaded lightly and the higher concentration of 3% shaded in the darker color. Oil was not artificially added to the background or control tanks, so these treatments didn't vary with concentration. My third objective is to determine if salinity affects the chemical structure. Half of my tanks were exposed to low salinity of 14, and half were exposed to standard estuarine salinity of 25. These values are in line with the mean daily salinity averages recorded in the summer of 2010 on the northeast side of Dolphin Island. Trace metal analysis was completed by separating the shells and soft tissues. They were dried. The shells were then sliced and mounted to a slide where transects were drawn perpendicular to lines of growth, and it was analyzed on the inductively coupled plasma mass spectrometer for these 10 elements. Tissues will similarly ground, pressed into a pellet, and analyzed for the same 10 elements. Preliminary analysis shows that zinc is the best indicator of oil exposure. Here I have shown my treatments on the x-axis and the concentration of zinc normalized to calcium on the y-axis. You will notice a consistent pattern where levels were high in the field, which was when oysters were initially under the care of Auburn Shellfish Lab and held in the lower part of Mobile Bay, as indicated by this lightest color. Once I received the oysters, they went through a 6-week acclimation period where you can see that the zinc levels dropped, as indicated by this copper color. Once the oysters were exposed to their respective treatments, the zinc levels once again rose, as indicated by this darkest color. There was a statistically significant difference between the acclimation period and the treatment period for all of my treatments except for spike. It is suspected that the reason for this is the spike oil may have been the most toxic, as indicated by lowest maximum growth. At this time, I don't have results for my control treatment. But you can see that zinc is efficiently assimilated by oysters once they are exposed to oil. Interestingly, there is evidence that zinc is high in the background water of Mobile Bay, as indicated both by high levels during the field period, as well as by the high levels in the background treatment period. It is important to note that this data represents 5 individual oysters. I still need to process replicate samples to confirm these preliminary results. Of my treatments, the Macondo crude oil showed the most noticeable results. Zinc is not shown here, but the rest of my elements are, and you can see the same general pattern of high levels in the field, a drop during the acclimation, and an increase during the treatment periods. there was a statistically significant difference for chromium, copper, and nickel. Other treatments only showed a statistically significant difference for zinc. The crude oil is the only one that showed it for the chromium, copper, and nickel. For Chapter 2, I will use organic compounds to confirm oil consumption, which will support the use of inorganic tracers discussed in Chapter 1. My first objective is to look at carbon-13 and nitrogen-15 stable isotope ratios as independent indicators of oil consumption. Shells will be separated with the tissue and shell, dried, ground, and packed for stable isotope analysis on the isotope ratio mass spectrometer at the UC Davis Stable Isotope Facility. My second objective is to use compound-specific stable isotope analysis, which allows me to look at specific analytes, and in my case, I will be looking at N-alkanes. This will help me to distinguish carbon sources which are not easily distinguished using traditional bulk stable isotope analysis. Again, samples will be processed at the UC Davis Stable Isotope Facility. Samples will first be dissolved in hexane. They will then be separated using gas chromatography into their individual hydrocarbon components, converted to CO2, and then analyzed for each specific N-alkane on the isotope ratio mass spec. Growth, survival, and condition indices are used to relate trace metal signatures to periods of growth. Survival was recorded daily. Growth and condition indices were measured for 10 to 15 oysters per treatment every 4 weeks. Growth is determined by measuring the longest length. Condition indices is a measure of oyster fatness, and there are many different ways to calculate this. I will use 3 widely accepted methods shown here, all of which involve measuring the wet and dry weights of the whole oyster, the shell, and the tissue. For Chapter 3, I will use feces and pseudofeces to trace the path of oil that is not assimilated by the oysters. Oysters were placed in petri dishes and allowed to clear their gut. I then collected the feces and vacuum filtered them onto a glass fiber filter and analyzed them for trace metals and stable isotopes as previously discussed. Pseudofeces was collected opportunistically and similarly vacuum filtered onto a filter and analyzed for trace metals and stable isotope analysis. My fourth chapter is to use calibrated elemental signature from Chapter 1, oysters, and compare that to oysters that were exposed to oil during the Deepwater Horizon oil spill. My first objective is to use the Chapter 1 oysters that have this calibrated elemental signature and compare that to oysters collected from TNC restored reefs along the Louisiana, Mississippi, and Alabama coast. These oysters were collected after the Deepwater Horizon oil spill. My second objective is to use the same calibrated elemental signature from Chapter 1 and compare it to oysters that were transplanted along the Mississippi and Alabama coast in May of 2010, which is when the oil spill first began. These oysters were collected in July and August of 2010, which was the end of the oil spill and just after the oil spill was capped. My third objective is to use these oysters as bioindicators to determine what regions were impacted by oil. This map shows the sample sites with the TNC restored reefs that were sampled in circles and the transplant sites shown in triangles. In conclusion, this method of using inorganic trace metal signature in oyster shells after exposure to oil may provide an effective way for determining historical exposure where studies were limited following the oil spill event. This method will complement long-term monitoring programs like the Muscle Watch program that currently analyzes soft tissues and will enhance reliability and accuracy. The ability to use shells, which store the information of both current and historical exposure, could save a tremendous amount of time and money for these long-term monitoring programs. I would like to thank my funding sources, the NOAA Restore Science program, my committee members, Dr. Carmichael, Dr. Kraus, and Dr. Appetti, as well as all the members of my working group, and the entire Carmichael lab, specifically Kayla, as well as many other people throughout the Dauphin Island Sea Lab that have helped me. Specifically for data analysis assistance, I received help from Ariel and Carl. Thanks for listening. Do you have any questions?
So,
yes.
I guess I'll start off. In looking at the elements that sort of are absorbed by these oysters,
Yes.
some were significant, some weren't.
Right.
are there expectations as to why some elements would be taken up more readily than others? What is it about an oyster that's going to take chromium and hold that and get rid of the other?
There are lots of different factors that can affect that. Some of these elements are useful for biological means, and so obviously, they're naturally going to take those up more than others. But there is evidence that the oysters do assimilate elements that aren't being used for any known biological processes. And exactly why they're choosing to assimilate those isn't really known, but there is evidence that they are. So all of the elements that I have selected were selected because either they are known to be typically in oils, and exactly what the elements are in different oils varies among oils, so you have to look at the oil specifically before you can use this calibrated signature if applying it to like a different spill outside of the Deepwater Horizon oil spill. And the other reason I selected the trace metals I selected is because they're known to be assimilated by the oysters. So all of the 10 I have selected either are known to assimilate by the oysters or are known to be in oil or both.
Is there any reason why, is there something about the chemical compounds that sort of make those things more assimilatable than other things, or is that just kind of an open area?
It's kind of an open area. Like I said, it's very species specific, and even like within salinity, there's variation. So some are assimilated better at lower salinities and others are assimilated better at higher salinities. Zinc doesn't seem to vary with salinity as much as others. It's just very unique to the individual element. Yes, Dr. Dorgan.
So can you go back to your graph with it was the zinc, the one where you had a bunch of fields?
That one?
Yeah. So you've got a lot of variability in your field collected samples.
Mm-hmm.
Do you know what?
Yes, I don't specifically know. There is a lot of variability, and what this doesn't show is this is, like I said, showing individual oysters, and I have been able to analyze 2. I plan to do more replicates than that, and so even within this, there is just a lot of variability among the oysters. We saw that same variability in like their growth, within an individual tank, so an individual treatment. There was lots of variability in growth, lots of variability in what they assimilated. So it's certainly the type of project that will require lots of replicates to see if there is a consistent pattern. And at this time, I don't have the data for the replicates to confirm. So like I said, this is preliminary results. Alex?
Your chapter 3, that's
The feces and pseudofeces?
those are from lab rates?
Yes. Mm-hmm.
So like fate, kind of, are we looking at any microbial degradation of the feces? Fate of those toxins that are taken up? It's just like, that just seems like it's, all right, this is how much is assimilated, this is how much is excreted, but that's not necessarily the end game, right? So there's another, are you looking at any?
So I guess, yeah, I should have clarified. We're really looking at the fate within the oysters. So the feces will be a combination of both what's being turned over within the shell, within the tissue, as well as what was never assimilated. And so that will not be able to be pieced out from my project. But what this will tell me is if it's being extracted as pseudofeces. So it's never actually going into the oysters and going into the gut and having the opportunity to be assimilated because the oysters are extracting that out. And that's what I'm really looking at with chapter 3.
So it's still not like proportions of how much spike oil and how much of whatever contaminant should be in there. It's assimilated, excreted.
Yeah, somewhat, and just kind of seeing it. This is semi-quantitative, so we're really just looking for patterns and trends of, is this an element that the oysters are primarily selecting to not even send through their gut, or they are assimilating well, or it's going through the gut but leaving as feces and just never being assimilated. Erin.
Okay, I don't really know anything about oyster growth. What's the turnover for like how often do they like replace or add another level layer to their shell? Or using their shells in terms of going back in time.
That, again, varies with a lot of environmental factors. So, like temperature, they're going to grow more during the summer when the temperatures are warmer. They're going to grow better generally at higher salinities. Obviously, there's a cap to that. Yeah, so, it's not even yearly. It's much more than that. So my experiment lasted for 16 weeks, and I saw significant growth and layers laid down during that time of 16 weeks. But I also used, it changes also with age. So I use juvenile oysters because they grow faster than adult oysters, right? So the layers would be slower in an adult oyster that's growing very slowly. And because I needed to get those layers, I chose to use juvenile oysters.
So when you go back and looking maybe back in time, so when you slice your oysters, are you going to find the band and collate that?
So oysters aren't quite as easy as mussels in terms of laying down in bands. But what is seeming to be the most reliable method of doing that is that strontium and barium are known to vary with salinity. And so because when I first brought them in, I had to acclimate them to the correct salinity, the salinity varied, and therefore I saw shifts in the strontium and barium. So I'm able to correlate that point where that strontium and barium shift was to where the field and the acclimation period ended. And then I'm looking for a similar type of signature that would indicate that it's oil, and that's where the oil exposure started. Yes.
So, how useful do you think a marker like zinc would be? Because I'd assume that there's lots of zinc in the environment. Other sources.
Right.
Paints on marine paints have a lot of zinc in them. How useful?
Right. Well, yeah, and that's, like I mentioned, is demonstrated here with seeing it high in the background as well as high in the treatment for the Mobile Bay water. So ideally, obviously, what we're seeing in the treatment signature is oysters that were exposed to that acclimation water that had low zinc. So we're seeing this shift up when they were in water that didn't have high zinc, so that zinc is coming from the oil. If they're out in the field and there's oil, potentially now there's even higher levels of zinc. So potentially we would be able to see that spike, but that would take studies beyond the scope of my project to determine. And at some point there would probably reach a maximum level of what the oysters can accumulate. So if the background levels are so high that they're already at that maximum in a given system, that would make it a less effective tracer than a system that had lower zinc background to begin with.
Okay, thank you. All right, Ed is up next. Ed is going to be talking about snapper.
All right, can everybody hear me? All right, cool. All right, hi everyone. My name is Ed Kim. Quick intro for those of you who don't know me. I'm originally from Philadelphia. I graduated from college there in 2016 and became an intern in Powers Lab in spring of 2017. From there, I became a technician, and just recently, this past fall, I started my master's. And today, I'll be presenting my prospectus to you, which comes in two parts. It's going to be a distribution and abundance, as well as an age and growth study of gray snapper, Lachenis griseus, in the north central Gulf of Mexico. Life history on these guys. They're more commonly known as mangrove snapper among recreational anglers, and they have a fairly broad distribution. They're located here in the Gulf of Mexico, all the way down to the northern reaches of South America. They're a fairly long-lived fish at around 25 years, maximum total length of around 90 centimeters, which is about 35 inches, and a maximum recorded weight of 20 kilograms, which is about 40 pounds. But here in the Gulf, you'll tend to see them max out at around 15, 20 pounds. These guys are broadcasting aggregating spawners, which means that, especially in the summertime, they'll group together on offshore reefs, release their larvae, and then those larvae will recruit back inshore to coastal places such as mangroves or seagrass beds. They'll spend a couple years there growing up, and then they'll finally move back out to the offshore reefs to complete their life cycle. These guys are also a popular recreational fishery, especially over in Florida, where they have head boats and recreational charters that will specifically go out for these guys. And I say Florida because that's where most of the population in the Gulf of Mexico is located. They're more sparsely populated here in Alabama. And actually, when I was an intern, I didn't even know that these fish lived here. This picture has a bit of a cool backstory to it. So on my second day as an intern, we're hook and line sampling in the Theodore Industrial Canal. And I pull up this guy, and I'm thinking to myself, what the heck is a subtropical snapper doing in the middle of a cold snap in March in 50-degree water? So I did a bit of super scientific digging around on the internet, and I actually found that there are fairly common species here in Alabama right now, but what I've underlined in red here is personal accounts from people such as recreational anglers and reporters who recall that not having really seen them previously, but now seeing that they're more abundant these days. And what they're hinting at is that with warming, you're starting to see a change in abundance. So this is a bit of hard data to support that fact. What I have here in the middle is a figure I took from a paper that was actually co-authored by Dr. Power and Dr. Heck, along with Joel Fraudry up at UNC. But what it is it's looking at Alabama seagrass bed trawling data from the 1970s and comparing that with more recent trawling data in the 2000s. And what they found was that there are species that have popped up that you have never seen before, as well as species such as the gray snapper that I've highlighted here that have been here in low densities, but then you start to see for them a nearly 105-fold increase in relative abundance, at least for the juveniles on the seagrass beds. And then over here to the far side of the slide, I have another figure from a paper that's looking at a change in distribution under a future warming scenario. So again, you'll see the most of them are here on the west coast of Florida right now, but under this current warming regime, you'll start to see them shifting more northwards as well as westwards. So this is just recreational and commercial harvest by region. I'm going to focus mostly on this figure up top here. You'll see that there is currently a fishery for gray snapper here in Alabama, but it's a fraction of what you would see in Florida, again, kind of reflecting that most of that population is in Florida. But again, I've also did this for a Gulf-wide total for all states and found that by far red snapper is the king, but gray snapper come in at second and are more than double the third most species in catch, which is the vermilion snapper, highlighting that this is in fact a valuable sport fish species in the Gulf. So for all the Gulf species, there's this process called CDAR, which is Southeast Data Assessment and Review through NOAA. And what it is it's a bunch of collaborators, researchers, and scientists coming together to discuss numbers and papers to determine a stock assessment for each species, which will then go into informing management. There was one in 2016 that designated gray snapper as a data moderate species, and what that means is that there was data to make a stock assessment. However, there are still clear gaps in knowledge that could be filled. So what I'm doing for my project is essentially looking at the research gaps in the north central Gulf of Mexico where we are at, specifically in the Alabama Artificial Reef Zone, which for those of you who don't know what that is it's a huge area offshore that's filled with artificial reefs that both the state as well as recreational anglers have put down to support biomassive reef fish species. In particular, what I'll be looking at is looking at their reef use as well as their habitat preferences in that zone, as well as an age and growth study, because age and growth studies have been done on this species before for the Gulf. However, most of those are skewed towards Louisiana fish and Florida fish, so they're highly underrepresented in age and growth studies in this region. So again, my first chapter is going to be on distribution and abundance. My chapter 1 objective is going to be evaluating the factors that affect the spatial distribution of them in the reef zone. And for these, I have 3 goals. One being the first being to map the spatial distribution of their nominal abundances. 2, to generate a standardized index in order to be able to estimate the relative abundances. And finally, to characterize the probability of encounter, or just that you would see one under a certain set of circumstances in the reef zone, which I'll explain later. So as far as data collection and processing goes, our lab conducts a paired vertical longline and ROV survey in the fall and spring of each year. What happens when we approach this reef is that we'll put the ROV down, sit on one side for 2 minutes, record it, sit on the other side for 2 minutes, record it, and then finally take an overhead 360 view from atop. What happens is I'll then take that video back to lab and look to see if there's gray snapper present at all. So there's gray snapper, there's red snapper. And after that, if they are present, then I'll look through the entire video, take 5 counts where I think they're the highest in abundance, and from those, I'll take the highest one and use that as my max min count, which is essentially just a conservative minimum estimate of fish on that reef. And this is going to be of relative abundance because you don't know if there's fish off to the side here, off the side here, or up here. And finally, I'll also be incorporating laser length measurements from the ROV into this chapter. So as far as mapping nominal abundance goes, what nominal abundance is just saying that's the raw max min count data in its purest form. This figure shows them mapped out already. This actually says total count. I'll be updating that with max min count, but it's essentially the same process where I'll be binning the values and looking at them all across the reef zone. And this includes things like different habitats, such as pyramid here, chicken coop here, natural reef structures, as well as year, depth strata. And the next steps in the process for me for this will then be to separate those out and see specifically how pyramids throughout the reef zone or chicken coops throughout the reef zone vary in abundances. After that, I'll be making a standardized index of relative abundance. And what that is I'll be using the nominal abundances, and the reason I'll be doing that is because it's a zero inflated data set, which means that I'll see gray snapper on a lot of these reefs. However, for a majority of them, I actually don't see them. So in order to be able to interpret this data, I'm going to have to standardize it using a delta log normal approach, which is a two-step process. The first being a binomial generalized linear model to see whether or not the snapper are there. And the second, if they are there, I'm going to be able to analyze the variability in that catch data. So for that, for the variability, I'll be looking at factors such as year, the season, the depth, and habitat to see how those affect the abundances. And finally, this is kind of still in the works, but I'll be making a probability of encounter analysis, like I said. That will be essentially saying what is the likelihood of seeing a fish on a pyramid versus seeing a fish on a chicken coop, as well as kind of predicting the degree of abundance that you'd expect. And the final part of this chapter is going to be looking at length measurements. So our ROV has two paired lasers that go out parallel straight like this. We know the length of those lasers, and back in the lab, I'll be able to measure the length of those lasers as well as the length of the fish and be able to come up an estimate for size. And again, I will also be looking at those by year, season, depth, and habitat. And the cool thing about this is that a NIMS facility over in Panama City, which is along the same latitudinal gradient, actually has a similar survey that they do except with natural reefs, whereas we have mostly artificial reefs here. So it'll be a cool comparison to look to see the differences in the length measurements between the natural versus the artificial reefs. So my chapter 2 is going to be on age and growth of this species, which is going to be important for the stock assessments because it'll be able to tell me things about its population structure and different size classes. So I have two main objectives for this one. The first will be to compare my own growth models, which I'll fit from my own data set, with regional Gulf of Mexico growth models, such as in Florida and Louisiana. And in order to obtain this objective, I'll essentially be describing age at length relationships, as well as weight at length relationships. And then for both of those, I'll be looking at them both sex-specific for male and female separated, and then pooled together. The second part of this is going to be evaluating the physical and reproductive condition of the population. And for sample collection, we do a lot of fishery-independent techniques in our lab, such as vertical longline and trawl, that I do have samples from. However, this is a really inefficient method of getting gray snapper because of gear bias. So most of my sample collection is actually going to be through fishery-dependent techniques. One of these is the deep sea fishing rodeo that most of you know about that occurs every summer here at Dauphin Island. And I've also talked to recreational charters such as the Escape and Lady Anne over at the marina to dockside sample from them as well. And then to supplement my sample size further, I'm going to be hook and line fishing as well as seining inshore near seagrass beds in the summer to obtain smaller juveniles. Currently, I have 396 otoliths backlog that I'll be looking at. However, I'll need a target of about 700 to 800 to have a robust sample size for my analysis. I have a standard protocol when it comes to looking at these fish. I will record 4 different lengths: the standard, the fork, natural, and stretched oil. I'll record weight as well as the sex of the fish. And for samples, I'll take otoliths for my aging study, and then I'll take gonads and intraperitoneal fat for assessing the condition of the fish. So this is what an otolith of a gray snapper looks like. But otoliths are really useful for aging studies because they lay down these concentric rings around a central core, and these are called annuli. But essentially what happens is that in periods of slower growth, such as during the winter when there's not as much food or it's colder out and their metabolism slows down, they'll start to lay down those bounds tighter together, which is what you see with these dark ones here. And then conversely, in the summer, when they have higher metabolism, when they're growing faster, you'll start to see these lighter bands, which shows you that those bands are starting to spread out a bit more. But this is an 18-year-old fish. I've put the red dots here to show where the bands are, and it has a margin code of 4, meaning a margin code essentially tells you how much of that next annuli has been put down. So margin code of 4 means that at least two-thirds of that next ring has been put down. Here are just some of the equations I'll be using to model my length at age and weight at length relationships. For the length at age relationships, I'll be utilizing 3 different ones, von Bertalanffy, Gompertz, and Logistic, and I'll looking to see the utility of each one and fitting them to my data set, and then using an AIC index to determine which is the best fit. For all of these, I'll be describing the growth parameters. So L infinity is the length at which growth approaches 0. So if you look at this curve up top, that would be where it starts to asymptote off. K for the von Bert is just a growth coefficient, and then T sub naught is the age at which length is equal to 0. And then for the Gompertz and Logistic, I have G being equal to growth coefficients, and the alpha is just that inflection point within the curve. And then for the weight length relationship, I'll fit that to a power function, which is typical. And for all of these, again, I'll pool them by sex and also look them male and female to determine if there are any differential growth rates due to sex. The last part of this chapter, I'll be looking at population condition with 3 factors. The first being a Fulton condition factor, which is essentially looking at the weight of the fish versus its size. And the underlying assumption here is that a heavier fish is a healthier fish. And then I'll kind of couple this with the gonadosomatic index, also known as GSI, which is just looking at the ratio of the gonad weight to the body weight. And this is telling you the relative allocation of growth towards reproductive growth, so the gonads versus somatic growth, which is the body weight. And using these two, you can kind of tell when these fish are starting to spawn. So right before they start to spawn, you can tell because the GSI will be relatively high because they're allocating more energy there, and the Fulton condition factor will be somewhat low. And then you can tell after they spawn, GSI will be low, but then Fulton condition factor will start to increase. And then alongside this, I'll also be sampling intraperitoneal fat, or IPF, and that's just another measure of energy storage versus energy expenditure. So finally, just to wrap things up, the utility of this project is that it's the first independent study of its kind on distribution and abundance, as well as age and growth in North Central Gulf of Mexico. And this is really important because it provides the basic life history and characteristics of a species that's necessary for an assessment. So this will go into supplementing the existing CDAR assessment, which actually occurred last year, which is how I found out these gaps in knowledge. And then also I'll be able to compare my own models against those in other regions and see how gray snapper here are different than populations elsewhere in the Gulf. And a few acknowledgments. I'd like to thank Alabama MRD, NIFWF, and CMAP for funding that allows this project to be possible. My advisor, Dr. Sean Powers, as well as our lab manager, Crystal Hightower, who spent countless, probably years by now, offshore using the ROV. And then CAM's Mike and Skipper Theory for dockside sampling, as well as Dr. Kelly Darnell and Christian Hayes over at GCRL for providing samples from Mississippi. With that, I can take any questions. Yes?
The comparison to existing growth models,
Yeah.
how old are those existing ones? Because one thought I was just wondering.
One was conducted, I believe it was for Florida, was in 2008. There was another one for Louisiana. I'm blanking on the date for that one, but they are I believe, within a decade of now.
I'm just thinking, these things could be changing as well. So if you see differences between your growth models and the previous ones, it might be a spatial pattern, but it could also be that growth has changed in the last decade.
Yeah, those are the most recent ones that I know of. I've done a lot of digging to see if there are any others, but it's mainly for Louisiana and Florida. And then they did use those 2 studies for their CDAR assessment, which was just last year. So I'll be updating those with mine for this year, and hopefully I'll be able to see if there are any comparisons between the 2. Any other questions? Yes?
So do you have any hypotheses, like, there's been a lot of work done on red snapper, like how do you expect gray snapper to be similar or different, or can you look at differences in red snapper between the different geographic locations and predict something about what you'll find?
In terms of their abundances with red snapper? Or
the age group curves, are they similar? Or,
I actually have not investigated that too much, so I personally don't know that. I can kind of talk about abundances if that would help. But so this is kind of in the works as well. But one thing I'm thinking about looking into, I'm not sure if I'm going to include it yet, is that offshore on these reefs, I do a lot of ROV processing in the lab. So like I just completed a year a couple of weeks ago what I've noticed is that on certain reefs, you'll tend to see a lot of gray snapper, but not a lot of red snapper. And then conversely, when you see a lot of red snapper, you don't see as many gray snapper. So I'm kind of interested in looking at spatial variation and seeing if there's any sort of in situ competitive effects on certain reefs, perhaps by depth strata, by reef types. But that's something that I'm looking into. Yeah, so I can't comment too much on the age and growth part of it, but regarding red snapper, that's something that I'm potentially looking into. Yeah.
Okay, it's attached to you.
Next here, Merritt, McCall. We're going back to the oyster well.
It's a good mix between the two. All right, can y'all hear me? Yes, no, maybe? Okay. Get this attached. Awesome. All right, so my name is Merritt. I am from Austin, Texas. I completed my undergraduate degree at Auburn University, and I am now here as a master's student in the Powers Lab. Today I'm going to talk to you about the importance of oyster reef design and setting in restoration success in Mobile Bay. So I'm going to start with the eastern oyster. This oyster is the most common oyster found here in Mobile Bay. It's a marine bivalve, but it also spans the entire eastern seaboard. So it's found all the way north in the Atlantic, and even down into South America as well. Oysters in general provide valuable ecosystem services. They protect the shoreline from wave damage. They also will filter water, and provide valuable habitat and a food source for marine species. There's a great commercial importance of this oyster, the eastern oyster in Alabama. They are very important to an important fishery here, and I believe that it's in top 5 landings as weight in Alabama fisheries. So because of that, we have a ton of restoration that's going on to restore this ecosystem to what it used to be with those wild oysters, just because they're so incredibly important. Kevin Gregalis was a master's student in the Powers Lab a few years ago, and he published a paper along with Dr. Sean Powers and Dr. Ken Heck looking at the restoration of oyster reefs along a biophysical gradient here in Mobile Bay. It was a short-term restoration analysis, and they concluded that abundance is dependent on this biophysical gradient that they had set up. They looked at they also concluded with oyster mortality that oyster drills, the southern oyster drill, is an important predator on these oyster reefs, and looking at all of this through oyster survivorship on the reefs. And that is going to be the number what I'm focusing on with my work. So these are the reefs that they deployed in 2004. Our first reef is Cedar Point. That reef is just north of Dolphin Island, this one right here. And it has it's on the western side of the bay, so it has that awesome oxygenation, and as well as a history of natural oyster reef. Our second reef was Sand Reef, so the one right here next to Dolphin Island. That has that oxygenation factor. However, oysters were not historically found here. And our third reef is Shellbank over here on the eastern side, which does experience those high hypoxic conditions, and there is no history of oysters present. So in order to collect samples, sessile invertebrates, they dove and did oyster quadrats. So they took a 1/4 meter squared quadrat, placed it on these oyster reefs. These reefs are 25 meters by 25 meters. And if we actually go back, you can see that there's 8 reefs located at each site. So 4 high relief reefs, which are about 1 meter off the bottom when they were deployed, and the low relief reefs being 10 centimeters off the bottom. So they looked at each reef. They're taking a sample from both the crest, which is the top of that reef, and the base at the bottom of that reef. Those samples were then taken back to the lab and processed, and then the data was extrapolated to assume that that 1/4 meter was representative of 1 meter squared on that reef. Bathymetric surveys were also done in January of 2006 because these reefs were deployed in 2004 prior to Hurricanes Ivan, Dennis, and Katrina, just to make sure that they were still there looking good. So they found that Cedar Point did have a significantly greater abundance of live oysters, and that is the well-oxygenated reef with a history of wild oysters present. And they confirmed that the biophysical gradient does exist when it comes to oyster abundance. So that takes you to my first chapter, which is just an evaluation of this restoration. So I want to see if after 15 years of these reefs being deployed, if that biophysical gradient will still exist among these sites. I want to also look into relief types, so looking at high relief versus low relief, seeing how that's going to impact abundance. Also looking at sample location on that particular reef. So looking at a high relief reef, what's going to be better, crest versus base samples, where are more oysters going to be present. And then also looking at it over the years. So we have lots of samples because this has been a monitored project. So looking at the yearly variation. And I want to figure out if this project was successful long term. So in order to do that, I'm working on analyzing the previous data with a focus on the live oyster density. So lots of other samples were taken, including live oyster density, but that's going to be what I'm focusing on. And live oysters, we are considering those oysters greater than 30 millimeters. So from 2005 to 2008, we had samples taken in both the spring and the fall. In 2009 to 2013, we had fall-only samples. And I plan on going out this summer to perform oyster quadrats once again, using those same sampling methods, and then also hopefully getting a side scan survey out there to just assess what's going on with these reefs. It's been 15 years, sediment moves and changes, so what do these reefs look like today? So far, I've just been using some of the data that I have, and I have found that when looking at live adult densities, mean densities, the high relief reefs, which are the lighter green, the densities are significantly greater on those reefs versus your low relief reefs at each site. This also can be seen when you look at crest versus base. So looking at all of the crest samples, you see a significant difference than looking at all the base samples at each site. So that's very interesting, and I'm excited to see if that is something that carries on into this year's sampling. And then also looking at the sampling looking at just the mean oyster density at each season, so spring, fall, whenever we are taking those samples. The first thing that jumps out to you is the spring of 2006. So this was about 2.5 years after these reefs were deployed, and we had a great mean density abundance here. And then the other thing that kind of jumps out at me when looking at this is that fall of 2008. This is when there was a record drought here in Alabama, and so we think that the increased salinity didn't directly affect the oysters, but allowed for oyster drills to move into the area, causing deaths with that. And so taking those oyster drills, I'm actually going to use that for my second chapter, looking at a predator-prey manipulative experiment in that environment. So there's lots of predator-prey interactions that do occur on these oyster reefs, and I'm only taking a very small sample of that, of those interactions. So I'm going to be using, my sample fish is a sheep's head. They're a generalist predator on oyster reef environments. And then looking at the southern oyster gill, I had Ed help me out with some collection here. And then using stone crabs, and of course the eastern oyster. So I am using seed size oysters, so they're 1 to 3 inches. And and then pretty much any fish that I could grab. So when looking at this experiment, I want to see what trophic interactions are occurring in the system, see what's going on with these different species in there. And I want to see if the sheep's head being present, if that would deter oyster gill predation at all. And then also, oyster gills don't have a natural predator, so looking to see if sheepshead are going to consume it. It hasn't been documented in any of the feeding studies on sheepshead prior to this, but just seeing kind of what's going to happen there. So I had to collect all of my samples to begin with. We did hook and line sampling for the sheepshead out on the rigs, and as you can see, this is a live well that I have set up in the back of the Nancy. I have 4 different fish in there, and this was just to get them back here to the wet lab. I am getting my oysters from Auburn Shellfish Lab, and they're providing me with crabs that are coming in their oyster bags as another source of food for those sheepshead during these trials. And then these oyster gills we got off docks. So after everything was been collected, it was acclimatized to my experiment. Then everything was randomly assigned to treatments. I am currently in the middle of my first 5-day trial. It ends tomorrow. I'm really excited to see what happens. And then at the end of all of that 5 days, I'm going to be sacrificing my animals and looking at them for analysis. So I had to set up my mesocosms, and so my fish were first living in the wet lab, and then we were moved to the mesocosms. As you can see, they really, really love these cinder blocks. I picked that cinder block up and down, and that fish stayed right there the entire time. But oyster reefs were placed in my mesocosms. You can see pictured here. That's just a cinder block covered in Vexar with a market-sized shell zip tied onto that Vexar to mimic an oyster reef environment. If the treatment calls for live oysters, those live oysters were placed on top of that oyster reef, mimic oyster reef. I also have 100 micron filters placed on all of my tanks just to keep some of the sediment out to allow these oysters to survive. So I have 5 different treatments. This is my first one. It has a sheepshead, oyster gills, and oysters in it. Stone crabs are found in all treatments as a alternate source of prey for the sheepshead. The second treatment has sheepshead and oysters in it. And then my third one being sheepshead and drills. Number 4 is drills and oysters. And then finally, I have just oysters. So this is just a little chart of kind of everything that I just showed you, but it shows you the numbers behind everything. So I'm going to have 5 replicate trials. If a sheepshead is present, it's just going to be 1. I have 50 live oysters in the tanks that do have live oysters. 6 oyster drills. Those are varying size. So I have 2 small, 2 medium, and 2 large in there. And then 8 stone crabs. So for data collection, I'm going to look at the stomach contents of the sheepshead, basically to see what they were eating during this trial period. And then I'm also going to be looking at survivorship of my oysters, oyster gills, and stone crabs, basically seeing what happened, are they still there? And with all of that, hopefully I will be able to provide some sort of picture as to what's going on these reefs. And with that, I would like to thank my advisor, Dr. Sean Powers, lab manager Crystal Hightower, and Pierce Cooper for helping me with experimental design, all the lab techs and everyone who has helped me set everything up, tech support for getting those tanks up and running because that's not an easy task, Brian Jones, and then Captain Jason Gamble as well. And with that, I'll take any questions. Yes. Right.
Are you looking at like restoration type from all these like restored areas?
I am not, because all of these reefs were all placed out, and they're all concrete rubble.
They're made of concrete?
I believe so, yes. And so just looking at those reefs in particular, not necessarily other reefs around the area. Sheepshead are known to eat pretty much anything and everything. They're generalists, but they love crustaceans. And so I honestly am not entirely sure what's going to happen quite yet. I'm hoping that the presence, if the sheep's head aren't consuming those drills, that them rooting around in those oysters looking for things is going to, mess up drill behavior so that they're not able to predate on those oysters. But I'm not entirely sure just yet. Yeah.
What do you know about the abundance of the other organisms you're going to include in the MESOCOSM studies in your field sites? Are they really parasite-killing oyster drills or
Yeah, so sheep's head, they don't stay on oyster reefs. They move through to and go offshore to spawn. But most of the time they're in the bay and all that. They love structure and stuff like oyster reef environment. Oyster drills also vary with salinity. So oyster drills do not like low salinity. And right now, this low salinity, it's really hard to find them in the wild. They move in generally when the salinity is higher. So yeah, there's kind of a vary, but there is an overlap of all of these species on a particular reef.